day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Vedika Maraj, and this lesson is brought to you by the Gauteng Department of Education in collaboration with Sibona Discovery Center. Today we are going to be doing genetics and inheritance, the last little bit of genetics and inheritance, which is called mutations and genetic engineering. But I'm not alone today. I'm here with my colleague and my co-presenter, Tiniko Vuma, who will also be presenting together with me. So let's get right into it with our lesson objectives. At the end of this lesson, you should be able to do the following. You should be able to define a mutation, differentiate between gene mutation and chromosomal mutations, and you need to know examples of them. Describe the effects of mutations, and there are different effects. There are harmful ones, you get harmless ones, and useful mutations. Describe how mutations contribute to genetic variation. Define biotechnology and differentiate between biotechnology and genetic engineering. Stem cell research, genetically modified organisms, cloning, and you should be able to describe and apply the use of blood groups and DNA profiles. And lastly, describe the mutations in mitochondrial DNA used in tracing female ancestry. Right, so before we go into mutations, I just want to pause here and do a little bit of like a pre-knowledge or a baseline of what information do you need to know before we actually go deeper into mutations. So I want to remind you of the DNA structure. So you know that in the DNA structure we have nucleotides, the basic units of our nucleic acid are nucleotides in the DNA structure as well as in the RNA. But let's look at the DNA itself. So in the DNA we have the nucleotides, as I said, but what can happen is we can have mistakes or errors that occur in that DNA sequence. So the nitrogenous bases in each nucleotide, the sequence of those nitrogenous bases is going to determine whichever code that DNA structure is going to be coding for whichever protein it's going to be coding for. So if there is a change in one of those or two of those or three or any of those nitrogenous bases, it changes the sequence of that code. Okay, so that is a mutation that you would have already known about. Okay, you did it when you were doing our first topic for this year, when we did DNA, the code of life, and we were looking at protein synthesis and DNA replication, where we saw that these errors may occur. Now, so that's the background knowledge. Okay, so we are not talking about a different type of mutation here. We are just expanding on that knowledge that you already know of our mutations. And we're going to go more into detail about the different types of mutations that we have. But before we go into anything, you know that we always have to do terminology. So grade 12s, I just want to stop here again and also emphasize the importance of terminology. It's very important that you go through all of the terms before you're starting a new topic or a new subtopic. And it's important that every day you revise on these terms, because your teacher is going to build the lesson up in every lesson, and you need to know the terms, otherwise you're not going to understand. And then if you don't understand the one lesson, then you are going to not understand the whole topic. So please make sure you study for your weekly terminology test that your teacher should be giving you, or however your teacher does your terminology, but let's not forget about it. So for this section, you need to know what a mutation is, and it's a sudden change in the genetic composition of an organism. And then you get two different types. You get a gene mutation and a chromosomal mutation. So let's break the word up. So you already know the mutation is a sudden change in the genetic composition, right? And then the gene part is referring to the change in the sequence of the nitrogenous bases or the nucleotides in DNA. Okay? And then the chromosomal mutation is also when you have a change, but this is when you have a change in the structure or the number of the chromosome. Okay, let's go back a little bit to chromosomes. What do you know about chromosomes? You learned it the first time in grade 10. Then you saw it again in grade 12 when we did the DNA code of life, when we did genetics, when we did meiosis. Okay, this is where you know of the chromosome. So you know that we can have changes in the structure of the chromosome, and you can have changes in the number of chromosomes. Then we come to biotechnology. It's the use of biological processes, organisms, or systems to improve the quality of human life. And then genetic engineering is a process where scientists alter, swap, or manipulate the genes on the DNA to produce an organism with desirable characteristics. But we'll unpack all of these terms in the lesson today. Right, so like we said, we explained the difference between the mutation, right? This is just showing you another picture where it is showing you now the gene mutation. Here on the picture is showing you a normal gene that is going to be forming a normal protein. And then here it shows you a mutated gene. Why is this a mutated gene? This orange little nucleotide or nitrogenous base is showing us that there has been a mistake or an alteration. And that has changed. So what it can do is it can either now form an abnormal protein or no protein, or that change can actually not have an effect and the protein can just be the same protein that is formed. Now this is where I'm reminding you about DNA code of life when we did this in the DNA code of life. So remember we spoke about that. We said that sometimes, so you all know that DNA has the master code or has a code for production of or synthesis of a particular protein, okay? And it's because the tRNAs pick up specific amino acids, right? So if the nitrogenous base is going to change in the DNA, that might cause the mRNA codon to change, which will cause the tRNA to change. And if the tRNA changes and picks up a different amino acid compared to what was originally there, that can cause a different protein to form. But sometimes that change in the DNA structure doesn't have an effect. Why does it sometimes not have an effect? Because sometimes there are some tRNAs that are picking up different a different tRNA, that can pick up the same amino acid. So we can have two different tRNAs, but they are picking up the same amino acid. So that means the protein sequence will not change. A chromosomal mutation, on the other hand, is the one where we talk about the chromosome structure, okay? There's going to be another diagram where I'm going to explain that. But let's first look at the different types of mutations that we get, other than your chromosomal and your gene mutation, okay? Those are the ones that are affecting specifically your gene or your chromosome. But now you also get mutations that are harmful, harmless, and useful. Okay, so let's have a look at this table. So if we look at some harmful mutations, we are looking at it as a cause and effect manner and what is an example. So an example of a harmful mutation are genetic disorders. For example, hemophilia and color blindness. So what is the cause of this? What actually causes hemophilia and color blindness? So you learned about this when you were doing genetics, right? And if I want to remind you that these are sex-linked traits, okay? Which means that these disorders are occurring on the gonosomes, yes. So the allele for these mutations are carried on the X chromosome. So here, what is the cause? It's the change the DNA responsible for the production of a specific protein. What is the effect? It causes changes to the organism's physical appearance or functioning due to an incorrect protein being made. Okay, and that's how we come to the genetic disorder. Harmless mutations are, for example, freckles, blonde hair, and baldness. Yes, these are all mutations, okay, but they are harmless ones because freckles are just like these little dots that we have, like little bouts of sort of like pigmentation, darkened areas that some people may have. Blonde hair, you would know is that, you know, yellowy color or whitish color hair, and then baldness would be, you know, the lack of balding in some areas, having less hair. So the cause of this are the changes in the non-coding DNA, the DNA not involved in making proteins. Okay, so these ones are harmless and they don't affect the DNA that makes proteins. Not all of our DNA in our body are responsible for making proteins. And the effect of this is that it does not affect the structure or functioning of the organism. Useful mutations is, for example, natural selection. So you don't know much about natural selection yet. We are going to do it in term 3. I just want to put it in here, term 3, when we do evolution. But we can still explain it here. This is when there's a change in the DNA responsible for the production of a specific protein. So this is once again got to do with the proteins. Protein made can increase the organism's chance of survival, and when the gene is passed can lead to genetic variation. So what this is saying is that the protein can cause it's going to cause a specific mutation, but this mutation can actually help an organism to survive. Okay, because natural selection is all about changing and adapting to the environment. Okay, it's about random changes occurring in the body and in the DNA, which allow organisms to develop characteristics that's able to make them survive, or be able to, their chances of survival be higher. And then this gene can then be passed down, because what happens, as you know from human reproduction that you've done and from genetics, is that you get one chromosome from your mother and one chromosome from your father. So essentially you get 50% of your DNA from your mother and 50% from the father, and these are going to get passed down. Okay, and that can cause genetic variation. Okay, that we can be different now and not the same because of this mutation. So once again, this is an example of a gene mutation, which is in the change in the sequence of nitrogenous bases or nucleotides in DNA. It's very important that you remember these words: sequence, nitrogenous bases, or nucleotides, and that it's in the DNA. So it's hemophilia. You all know what hemophilia is, and then you also know about the other one that I spoke about, which is color blindness. Okay, so so let's go back to the hemophilia. It's the absence of the blood clotting factors, and this is all because of the wrong protein being formed. And color blindness is the absence of proteins that make up either the red or green cone or photoreceptors in the eye. So once again, it's the ones that are affecting these specific proteins. Then coming more on the chromosomal mutation that I was avoiding to speak about because I was waiting to come to this diagram. So you all remember this karyotype, right? We did it earlier this year. So we have this karyotype here, and it's showing us all the chromosomes in the human body in a cell. In a cell, and this is a human one, right? So it's all the chromosomes in one cell, and in this particular instance it's a diploid cell, okay, because they're all in homologous pairs, and as you can see we have 22 pairs. And let's just remind ourselves, how many autosomes do we have? We have 22 pairs, which are 44 autosomes. And how many gonosomes? 2. We have 2 gonosomes, or 1 pair. So this is showing us, like I said, it's a change in the structure or the number of chromosomes. And in this particular instance with Down syndrome, it is when we are having an extra number of chromosomes on chromosome pair 21, and we have an extra one there. But you can also have one less. Okay, even though we're not doing that in life sciences, it's not part of your examination guideline, but you need to consider that you can also have one less chromosome, and it can affect any of these pairs. You can also have one more on the gonosome. You can have a change in the structure. You just need to understand what we mean by a chromosomal mutation and then apply it to any question that the examiner can give you. You will see when we do some questions now. Right, so just to recap there quickly before we do the questions, you need to know the definition of a mutation, a gene mutation, and a chromosomal mutation. You need to know examples and the cause and effect of harmful mutations, harmless mutations, and useful mutations. And then you also obviously need to know how to apply it, okay, to different scenarios. So here we have our first question. A sickle cell disease is caused by a recessive allele and first appeared in humans as a result of a gene mutation. So they're telling us now this is a gene mutation. So now you must think in your mind, okay, what did ma'am just tell me about a gene mutation? Even though we're not learning about sickle cell disease, but they are telling us that it's caused by a recessive allele and it first appeared as a result of a gene mutation. So they are saying that there was a change in the sequence and the nitrogenous base sequence. Right. The table below shows the number of children born with sickle cell disease in some regions in a particular year. So these are the questions that we need to do. Define the term mutation. Right, so as we explained it, it's a change in the sequence of nitrogenous bases or nucleotides in a gene. The change in the sequence of the nitrogenous bases. You cannot just say it's a change in the sequence of DNA. Okay, because that can actually essentially mean anything really. It's the fact that the nitrogenous bases here and the nucleotides itself are affected. Which region had the highest number of children born with sickle cell disease in that year? So let's go and look what is the highest number over here. Right, it is Nigeria. Yes, it is Nigeria with 91,011. Okay, so that is a very high amount. It's the largest amount there. So as we can see, it's Nigeria. What percentage of the worldwide total of children born with sickle cell disease came from the Democratic Republic of Congo? Show all your calculations. They want to know what percentage came from Congo. So let's go to Congo. What is the amount there? This is the amount. This is the one we're going to use. And they want to know the percentage. So they want to know how many out of the total, right? So how many of the total? It was 39,746. That was how many came from Congo. And the total amount is 305,733. Okay, so what you need to do is you need to divide. You take the number of Congo, how many were in Congo, and you divide it by the total number and you times by 100, and that's how you get a percentage of 13%. Okay, grade 12, if you're not sure how to do a calculation, as soon as they're going to be asking you for a percentage, you know that somewhere it's going to be times 100. So even if you get this part wrong and you are unsure, please still put your times 100, because you will still get a mark there. So at least you can get 1 out of 3, even though you don't even know what to do. But because you realize that we're going to have to work at a percentage, you need to times by 100, you can get a mark. Use the letters D and D, capital D and small letter d, to give the genotype of a person who suffers from sickle cell disease. So they told us that it's caused by a recessive allele. Okay, and remember that in order for something to be phenotypically present, then the genotype would have to have 2 recessive alleles. So that would be small letter d, small letter d. For a person that carries the allele but does not suffer from the disease, then that means that they have the recessive allele, but because it's not, they don't suffer from it, so they don't have it phenotypically, their other allele must be a capital D. So grade 12, I want to mention something here to you. Can you see that, yes, there is some genetics here, but it's not your traditional. Can you look at this question? It's asking you a number of things. It told you some data handling questions, and then it asked you about a mutation, to define a gene mutation, but then it gave you some calculations and some data handling, and then we came back to the genetics again. So the tricky thing about this section, especially the gene mutations, is that you can see it throughout paper 2. That's the other thing I forgot to mention. This is still part of paper 2, but you can see it everywhere because you have it in DNA, right? You have it in meiosis with the Down syndrome. You can see it in evolution, right? And then you have it in genetics. So you can really see mutations throughout your question paper. So it can sneak up on you. So please be aware of that. The next question, the diagram below shows a stage in protein synthesis. You see, now this is the example where we are talking about the mutations within DNA. The table below shows the tRNA molecules that code for different amino acids. Right. In codon 3, guanine was replaced by cytosine as a result of a mutation. So let's go to codon 3. Let's look at our picture. We have 3 codons. Okay, so that means, so this is mRNA. It's very important that we analyze our diagrams, okay? Then we have the round circle is the ribosome, okay? Then we have A over here. It's joining the amino acids, so they are peptide bonds. And then we have B, which is the amino acid. And then just so that for understanding, this is our anticodon. Okay. So now they want to say in codon 3, so this is this one over here, guanine was replaced by cytosine. Okay? Explain how this mutation will affect the protein that will be formed. So the first thing you need to do is you need to tell us what is going to be happening. Okay? And I prefer the second one here to first tell us that UGA codes for threonine And UCA codes for serine. So here's serine. Okay, and you can see here that UGA, it codes for serine. Okay, and then the other one codes for a different protein. So threonine will be replaced by serine, forming a different protein. So because the amino acids are going to be different, so you just need to say which amino acids will change, and then it will obviously form a different protein. In this instance, it is going to form a different one. This is a different question now on the human male karyotype. Right, so here, let's have a look at what we have over here. You can see that there are 22 pairs, right? 23 pairs in total, 22 are autosomes, and these are the gonosomes. The gonosomes look normal. Everywhere we have 2, 2, 2, aha, except for this position here, number 16. Can you see something funny looks here? What's happening there? There is an extra chromosome. So what kind of mutation is this? It is a gene mutation, yes. How many chromosomes does this man have? So you can count them, but you can already see that we have an extra one, so this person would have 47. And how many chromosomes does a normal human have? A normal human has 46. And then it says this man has 3 chromosomes at number 16. This is a result of non-disjunction of chromosomes. What is this type of mutation called? It's called a chromosomal mutation. Why is it a chromosomal mutation? Because it is change in the number of chromosomes. Why is it not Down syndrome? Because this extra chromosome is not on pair number 21. So that's very important to notice, because when learners see this question, they just see non-disjunction and they see mutation and they think Down syndrome. But it can only be Down syndrome if it affects number 21. And this one is affecting number 16. Right, now let's read the extract. Now, as you can see, now we also have a different form here. We have an extract question. So we had a DNA question, we had data question, we had a protein synthesis one, and now we've got an extract. Okay, 12, whenever you have an extract question, you should always read it through once with understanding, and then you should read your questions and then go again and read the extract, because now the original reading is going to give you an idea of what the extract is about. When you read the questions and you go back, now you are reading with intention, because now you've read the questions and you know that you need to look for the answers. So let's read it. Researchers have discovered that members of a particular family have high bone density that may be caused by a gene mutation. High bone density reduces the risk of bone fractures. 20 members of the family had their bone density measured and DNA samples taken. 7 had high bone density. The high bone density occurred throughout their bodies, but especially in the spine and the hips. Once again, we want us to describe what a gene mutation is. This question has come up often, right? A change in the sequence of the nitrogenous bases or nucleotides of DNA. You should know that when I wake you up in the middle of the night, you must be able to tell me what's a gene mutation and a chromosomal mutation. Explain why it was necessary for the researchers to collect DNA samples. So it's an explain question. Why must they collect DNA samples? And the reason why is because to check for the gene mutation in the individuals who had high bone density. Okay, they need to collect them so that they can check for that gene mutation that those ones have. And then what is the effect of this mutation? Now this one, do you think it's a harmful one, a harmless one, or a useless one? What did they tell us? They told us that it reduces the risk of bone fractures. Isn't that a good thing? It reduces the risk of bone fractures. Therefore, yes, so that's the effect of the mutation. It produces high bone density. It reduces the risk of bone fractures. So you can also say that it is a useful mutation. Calculate the percentage of the family members who had normal bone density. Show all your workings. Okay, so they said 7 had high bone density. Right. So, 13. who had normal bone. They want to know how many had normal bone. So they're telling us that there were 7. Okay, so they took it, firstly they told us that the total is 20, that they had 20 members, and then they said that 7 had high bone density. So then we have to say 20 minus 13 is equal to 20 minus 7 is equal to 13, and that's where this 13 comes from, because we had 20 of them. Okay, and 7 of them had high bone density, so the other 13 had normal bone density. So 13 divided by the total of 20 times it by 100 equals to 65%. So once again, can you see that there are 2 questions here that have to do with directly with gene mutation, but the other questions are a little bit different. Right, then moving on to our last question before we go for a little break, and then we will be back with the rest of the presentation. In a certain country, 25,000 individuals were suffering from hemophilia between 2012 and 2020. Hemophilia is the genetic disorder that is caused by a mutation, right? Which mutation is it? Gene mutation, yes. The pie chart below shows the percentage of individuals with different levels of hemophilia in this country. So here we see there are high is 40%, low 35%, and moderate 25%. Explain the effect of this mutation on an individual. Okay, so now grade 12, hemophilia is one that you need to know. Okay, it's in the examination guideline. You need to know what hemophilia is. You need to know how it's caused. You need to know all the crosses that have to do with hemophilia, whether you get it in a pedigree diagram or a monohybrid cross, or even in a question with a pie chart. And you need to explain the effect. Explain question requires you to answer in a cause and effect. You must mention a cause, and then you must mention an effect. Okay, so what will this cause? What is the problem with this one? It is harmful. Okay, it is harmful. It prevents the clotting of blood, which is harmful, right? That's bad. It leads to excessive bleeding, internal bleeding, and swelling of the joints, okay, and bruising. So it's a problem, right? It can cause all of these issues. So you just need to explain to us what it is. If you had just says prevents the clotting of blood, leads to excessive bleeding, you would have gotten 2 marks. Calculate the number of individuals who were moderately affected by the hemophilia in this country. Now they want to know the number of individuals. And what did we get here of moderately? What did they give us? They gave us a percentage. So they gave us 25%. Okay? And they gave us a total number of individuals. And the total number of individuals was 25,000. So they want to know the number that was moderate, but they gave us a percentage. So 25% of the 225,000, that's what we need to work out. How much is 25% of that 25,000? So how do we work that out? We say 25 over 100, which is now simplifying our percentage, times it by 25,000. And we are going to get 6,250. Or you could have done it in a different way. Okay, mathematically you could have said 25% times 25,000 divided by 0.25 times 25,000, right? Or you could have said 25,000 divided by 4. Right, so those were the different ways, okay, that you could have done it. For me, the first one or the last one was the easiest, but we are all mathematically different. However you did it, you saw, as long as it's logical and you come to the answer, you will get the marks. Okay, so before we get into that, I just want to do a quick recap quickly of what we did. So we spoke about the importance of terminology. We spoke about gene mutations and chromosomal mutations and mutations in general, and how mutations can affect our lives. Okay, and particularly what you need to know for examinations and for tests is that mutations can be anywhere. They can ask it in any of your topics in paper 2. So please make sure that you don't get blindsided by it. You practice enough past paper questions on it. I think we really covered a number of them over here. And I also want to quickly double check something. Let's see if we're able to go back a bit. Okay, wait a second. Okay. When we come to this question on proteins and on protein synthesis, I want you to please have a look at the worksheet. Okay, and please have a look at this question in the worksheet and look at other questions like this. This is really a question that you can get often when it comes to the mutations. Okay, so the take-home message is that the gene mutations has got to do with the change in the proteins. That's what will make it harmful. Okay, and then we have the gene mutations, which is the one that is affecting our chromosome number. It is not only Down syndrome. And the gene mutations aren't only hemophilia and color blindness. Okay, so that is what I just wanted you to understand. We will quickly take a 5-minute break, and then we will be back with Tiniko Vuma, who will take you through genetic engineering to the end of the lesson. Please stay tuned to have a quick comfort break, and then we will be back.
Welcome back, grade 12s, and thank you, Mrs. Maharaj Vedika, for taking us through mutation. Interesting, isn't it, that we have got different types of mutations, and now we are going to be looking at genetic engineering. So, looking at genetic engineering, this is some of the information that was done by Ms. Maharaj. So we're moving forward to start with our genetic engineering. You did your calculation and that was interesting on your side. And thank you. So now, genetic engineering. This is a very wide topic. We are going to look at what we call biotechnology. If you look at the definition, you need to be very careful with definitions. The definition of biotechnology is the use of biological processes, organisms, or systems to improve the quality of human life. Then we have genetic engineering. Genetic engineering is a process. Now we're talking about a process. In other words, when we spoke about biotechnology, this is like the broader explanation. But now, genetic engineering is more specified field where scientists alter, swap, or manipulate the genes on the DNA to produce an organism with desirable characteristics. I want you to keep this in mind, desirable characteristics, because we are going to dwell more on that. But genetic engineering is now divided into subtopics. And the subtopics that we're going to look at are, we've got genetic modification, GMOs, which are called genetically modified organisms. We will also look at stem cells research, and we'll also look at cloning. These three are the ones that we are going to be looking deeper into. You can see the link from biotechnology, that biotechnology as the broad spectrum is giving us genetic engineering, and you can see that we do have genetic modification, and we've got stem cells research, and we have cloning. I want us to look at the advantages and disadvantages of genetic engineering. You know, in life, even not in life sciences only, we have got advantages and disadvantages of whatever happens around our lives. But now for this topic that we're talking about, the good side and also the bad side. Let us look at the advantages. The advantages are genetic engineering is good because it will help in the production of medication or resources in a cheaply way. So people can have access to resources, but then that is going to be cheaper. And also the control of pests. You see, when we speak about controlling of pests, pests are able to be controlled with specific genes inserted into a crop. We are talking about crops. There are also other organisms that can be manipulated. And another advantage is it uses specific genes to increase crop yields, which now gives us what we call food security. You have learned about food security in grade 11. So you know that once we have got increased crop yields, it means that whatever is harvested, it's enough, and people will have enough food to survive or to live with. Selecting genes to increase shelf life of plant products. If we speak about shelf life, I want you to remember the following, a very simplified example. So under normal circumstances, if you take tomatoes, you plant tomatoes and they are organic, and you take them and put them somewhere in your vegetable rack, they can maybe last for a week or two, they become overripe and they get spoiled. However, when genetic engineering takes place, when the manipulation takes place, you'll find that instead of these tomatoes having 2 weeks, they can now last on the shelf for how long? 4 weeks maybe, or even 6 weeks, which is now a great advantage for who? For the person that is making money, because they make money out of making these crops. The disadvantages, however, are that the process is expensive. Grade 12s, I want to remind you of something. Whenever you find in a question paper, they ask you about the disadvantage of any kind of process, just remember to mention this standing one. It is expensive, because under normal circumstances, processes and things that take place, they are expensive, and you just get the free mark. And another disadvantage is that it is not good because it interferes with nature, and therefore we can say this is immoral. If we're saying tomatoes are supposed to last only for 2 weeks, why should they be manipulated to last for 4 weeks? If nature says they must only last for this long, why are we saying we can now use our own manipulation to make it last longer? So it's immoral. And there are also potential health impacts, which I can also link it to the next disadvantage, which is saying that we are unsure of long-term effects. We really don't know. We might be happy with genetic engineering at the moment, but we don't know after 20 years, after 30 years, after whatever years, what is going to happen, which is going to have a negative impact. And new pathogens could be made for biological warfare. Once we speak about warfare, there is a problem. So if we've got new diseases-causing organisms, which are a new strain altogether, now it is going to be very difficult to control them. And they can resurface, or they can surface from genetic engineering processes. And very important, one standing disadvantage of this process is loss of biodiversity. Think of those organisms, think of those crops, think of those animals, think of those livestock that are not carrying the required or favored characteristics. Think of them being neglected, and then they will gradually become extinct. Then we have got the pool of the varied organisms. It's now minimized because of genetic engineering. Hence, we are saying that this can result in loss of biodiversity. Let us now look at one example of genetic engineering. Genetically modified organisms. Let us just look at what this is all about. Genetic engineering is used to alter the genome of a living cell for medical, industrial, or agricultural purposes. There are so many reasons that can lead to manipulation or changing of the genome. When we speak about the genome, we are referring to the full package of the genes that are found in an organism, the genetic footprint that is found in an organism. Now, if this is altered, it means that at the end of the day, we are going to have some benefits in agriculture, in industrial world, and also in the medical field or medical world. This results in a genetically modified organism, GMOs, or transgenic animal. What is a transgenic animal? This is an animal with DNA from more than one species. You will see that when genetic modification takes place, there will be a gene that is taken from a different organism, and this gene is inserted on one species. And at the end of the day, this is going to influence how this transgenic organism is going to react, so that whatever is desired by the scientists who are in charge of the engineering story, they get as an outcome. GMOs, or genetically modified organisms, are used to breed more productive crops or animals so that more food can be made. So we're looking at yields. We're talking about crop yields. The outcome must be more. If it's milk, we must have tons and tons or liters and liters of milk. If it's meat, it must be a lot of meat that comes as an outcome of genetic modification. It can also be used to produce drugs or hormones, e.g. insulin, which have fewer side effects and is cheaper. If I want to give you a background, which I'm gonna expand later as we look at the process itself of insulin making, you'll realize that it is easier now with this genetic engineering to get insulin, which was not the same way in the past, and this is giving us a better benefit. And GMOs are also used to infect cells to cure diseases, right? And we call this gene therapy, such as brain tumors and cystic fibrosis. So, as much as, yes, we spoke about the disadvantages, there's a lot of good things about genetic engineering, because you can see people that are having issues with cystic fibrosis, people that are having brain tumors, these are all health issues. The manipulation can assist these people to have maybe an expanded and extended lifespan from it, and we can have a production of very good yields at the end of the day, and the list is endless. Let us now look at stem cells. So we spoke about GMOs, which is genetically modified organisms. We are now coming to another example of genetic engineering, which is stem cells. What are stem cells? In life sciences, it is very important for you to remember terminology. You must be able to write terms. That is basically your section A, question 1.2 years, which is about biological terms. So it is very important for you, as you study each and every chapter, to know what mutation is, to know what stem cells are, to know what genetic engineering is, to know what biotechnology is. And on that note, normally there's a fine line of a difference, and learners do get confused. You must be careful to know what biotechnology is, so that if the description that is, or the definition that is given in the exam is about genetic engineering, you do not give biotechnology as your answer, but you give genetic engineering or vice versa. Be always extra careful. For now, we're looking at the definition of stem cells. Now, when we speak about stem cells, these are undifferentiated cells that could grow into any tissue in the body. I want you to mark undifferentiated, and I will speak about this later today. So if we speak about stem cells which are undifferentiated cells, it means that they can be influenced towards a specific direction. And once they are influenced into that specific direction, they will be able to be manipulated to become tissues, which can later become even glands. And remember, there's the sequencing and the levels of a cell, which is the smallest of functional unit of life. And now tissues are a group of cells that are having the same structure, and they perform the same function. And tissues together now, they give us organs, and organs, they give us different systems. You will learn about nervous system, you learn about endocrine system, etc., etc. And where do we get, or where are we likely to get stem cells? Stem cells are normally found from embryonic or embryos. We've got embryonic stem cells. I just want to mention something about some controversial issues that happen in some of the countries regarding this. Did you know that in some places people will really make this a business? To say that they fall pregnant and now the embryos are removed. The reason for removing the embryos is that the stem cells are there. So it's kind of a business where embryos are removed. So it's like some kind of, should I say, not miscarriage, because miscarriage is natural, but abortion. So they are aborted so that they can, the stem cells, they can be extracted from the embryos for all the uses that stem cells have in life. We can also get stem cells from bone marrow. I'm this one typical person. When I eat my chicken, I like to break the bones and I go inside and I suck the bone marrow. I'm just enjoying that. So I'm just giving you an example that the bone marrow that is found in the bones has got large numbers or large quantities, or because we cannot count the cells. Oh, we cannot count the cells, in all honesty. They are a lot, and they are very minute, very, very microscopic. And that's where we can get stem cells. So where do we get stem cells, good people? We get stem cells from bone marrow. We can also get them from embryonic or embryos. Just for your information, when a baby is born, there is an umbilical cord. We have learned about this in reproduction. The blood that is found on the umbilical cord, it's very important. For some countries, what they do, they will take the blood there, which has got a lot of those cells which are undifferentiated, and they harvest it. Just like, where we've got seed banks, or where you've got money banks, you will be smiling because money, we know about money and we all love money. But we have got different banks where your bloods can be harvested, or seeds in terms of crops can be kept for future use. So if there's a need later, if, let's say, for example, my umbilical cord bloods with those undifferentiated cells are kept somewhere, later in life, then I get sick. When I get sick, it means that there is at least a possibility of going back to that bank and draw out the undifferentiated stem cells from my own blood. And then this can be manipulated to help me from my medical challenges. This is so interesting. This is so interesting. Grade 12s. We are not learning all these things for books, but we are learning so that they help us in our daily lives. Once you have done life sciences, you later look like a medical doctor, I promise you. Yes, because we are scientists, we're learning a lot. So learn to remember. Yes, learn to pass and learn to remember, because you'll help yourself and you'll help the people around you later. The uses of stem cells are, they can be used to treat cancer, and specifically cancers like leukemia, which is blood cancer, and also used for replacing dead cells in the heart after a heart attack. When a person goes through a heart attack, there are cells that will die. And now stem cells can go and assist where those cells are dead, and naturally the body is not able to produce more of those cells. But now with genetic engineering, the stem cells will be manipulated to produce or to replace those cells that died, and somebody has a chance to live. Interesting, isn't it? It is interesting, I tell you. And stem cells are also used for growing skin tissue to treat burn victims. Sometimes when a person is burned, there are different degrees. They will tell you about, this is 93 degrees burn, so you can see that it's really bad. And in order to rescue those people with that severe burns, wounds and stuff, stem cells can now be manipulated to grow and make up the skin that was lost from the burns. And this is normally an unknown and uncomfortable experiences that some people go through. Stem cells are also used for growing nerve cells to treat spinal cord injuries and Parkinson's diseases. You will learn about all the spinal cord stories in nervous system. But I want to tell you that the nerve cells that are there, once they die, they die. So it's not easy for them to be replaced. But if there's a need for them to be replaced, so that a person is not suffering from, because a Parkinson's disease, it's more like a mental condition. So if stem cells are there, they are there to assist or to help people that are having spinal cord injuries, because once you have an injury on the spinal cord, what will happen? It means that you'll really become paralyzed, And I know somebody saying, as much as maybe we can use this as baseline, although we are not doing nervous system. Somebody's thinking spinal cord is the vertebral column, the set of bones that are found at the back to support us so that we are upright. No. The spinal cord is the bundle of nerves that are running from the head and they go down. They are protected by the bones around them. So I want you to remember that the bones that are around them, they are there to protect the spinal cord. But then there are uncomfortable circumstances where there can be a situation where somebody gets the spinal cord safe as it is to get an injury. That's when now stem cells come into the picture to assist remedy some of the problems. And stem cells are also used to growing pancreatic cells to treat sugar diabetes. We know that the pancreas is one organ that is responsible for production of insulin. The cells there are the cells that we have, the islets of Langerhans on the pancreas. They are the ones that are responsible for secreting insulin, which is now a hormone which will help to balance the sugar levels in the blood. It's not even Selati or Hullet sugar that we buy from the shops to enjoy with our tea in winter, but it is glucose, which is the blood sugar. Interesting, isn't it? I just want us to look at some of the examples of cells that can be manipulated to form after we have stem cells. So you can see we've got stem cells, and stem cells can be manipulated. Remember, they are undifferentiated. They can be manipulated to form cardiac cells. We earlier on spoke about the replacing of the damaged cells after a heart attack, and cardiac cells are the cells that are found on the heart. We also speak about the enterocytes. Enterocytes, these are the absorption cells that are found on the small intestines. So stem cells can be manipulated to become enterocytes in the small intestines. Fat cells, for example, as well. We know about fat cells. The stem cells can also be manipulated to become red blood cells or red blood corpuscles. And you know, red blood cells in our bodies, what do? They are the transport system, which is responsible for transporting oxygen so that our entire bodies have got a full supply of oxygen, which is needed because our bodies, our cells even, they are all alive, and they must be well taken care of with nutrition and also with oxygen. And we are also looking at chondrocytes. These are the type of cells that we find on our cartilages. I just spoke about eating chicken. So if you eat a drumstick, for example, you'll see there's a white cartilage part which is there, and we also have cartilages between our bones so that they prevent the friction. And these are now the chondrocytes, and these are specific cells that can be manipulated from stem cells as well. And we also have epithelial cells that everyone is familiar with. We spoke about nerve cells. Remember, we said stem cells can also be used for growing nerve cells. So nerve cells which we now get on our neurons, and neurons, it's about our nervous system. I want to, as I promised you, to speak a little bit about this, the blastocyst. This is something familiar. You have done human reproduction already, and you have learned about the sperm fusing with the ovum to form a diploid zygote. Yes, to form a diploid zygote. And you have learned about how this diploid zygote will start now to undergo mitosis. And as it undergoes mitosis, it starts to grow. And then when it grows, it will now become an embryo. No, it's now morula, and then it will become a blastocyst. So there is the blastocyst. So throughout this, I want you to have a clear mind about stem cells as being undifferentiated. Think in your mind. A sperm cell is microscopic, very small. Think of the ovum. very, very small, fused together. A zygote is still very small as well. But when mitosis is taking place, they start to multiply, and marula is that ball of cells, and then later it's a hollow ball of cells, which is a blastocyst. And this is now going to grow bigger and bigger. But can you see that the cells from a single very minute microscopic sperm cell and a single microscopic ovum, we end up having a fetus. Then later we have, when the baby is born, this baby has got all the expected organs. There's a small heart, the lungs are there, the limbs are there, the head and the brain is there. So can you see that from that very fragile stage of development of the blastocyst, differentiation will start to take place, where now we are going to have cells that are going to clump together, and they are differentiated, and they go and form the small liver for this little one that is forming inside mommy's womb, and that's a fetus. And then some will grow, and then they will form the limbs, and some will grow and they will form the digestive system, although the fetus is still dependent on the mother while it's still gestation period. So you can see where stem cells are coming into the picture. Something else, cloning. So just refresh our minds. We said genetic engineering, and it's having different umbrellas. We have already spoken about genetically modified organisms, GMOs. We then spoke about stem cells, but now let's look at cloning. Again, the story and the song remains the same. You need to know the definition in order to give us the term so that you get a mark. And our learners have got a challenge on this. That is why we keep emphasizing that you must remember to do your terminology. So cloning, what is cloning? Cloning is a natural or artificial process. This is a process of creating genetically identical copy of an organism. That is cloning. Genetically identical a copy of an organism. Don't think about reproduction, because when the sperm cell and the ovum fuse together, that fetus or the zygote is going to be carrying the genetic information from paternal and then also from maternal origin. Therefore, the baby that is born is not going to be genetically identical to the mother or the father. But now cloning, it's a process where you see exactly the same organism as the one that was cloned. I want us to look at a clear illustration. If you get this right, there's no way that you'll go wrong with cloning. But you need to understand, because examiners have got all the different tricks of asking questions that you have studied about, and you must be able to answer them. If we look at the illustration of cloning here, so I want you to listen very attentively, and please look at the illustration itself. So we have got two sheep. We have got sheep A and we have sheep B. Just keep a note on that. There are two, how many? Two female sheep. And then, during the cloning process, what will happen? From sheep B, an egg, this is an egg. Yummy. It really looks like a real egg when you fry eggs and you see the yolk, which is yellow, which is very nice. You see the yellow part there, it's an egg yolk. And now the egg cell is taken from sheep B. What is taken is the egg cell. And the egg cell is a gamete, we know. It's a female gamete. And you know that if we speak about the number of chromosomes in an egg cell, is it haploid or diploid? It is haploid. So the egg cell will have half the number of chromosomes. I want to remember this as I take you through the process of cloning. Then the egg cell is removed from sheep B. And what happens? The nucleus is removed. And what happens to it? Thrown away. That is why, look at this egg now. This egg does not have the yellow that was there. The yellow is representing for us the nucleus. And the nucleus, you know, is carrying what? Genetic information. But the nucleus is removed and thrown away. Look at now sheep A. Sheep A, the body cell taken from sheep A. So if we speak about the body cell, I want you to remember body cell, somatic cell. Somatic cell, is it haploid or diploid? It is diploid, having a full or double the number of chromosomes. So any body cell can be taken from sheep A. And DNA is extracted. This is our DNA that is extracted. Where is the DNA found? It is definitely found on the nucleus. So that is removed from the somatic or the body cell of sheep A. And what happens is that now DNA from sheep A fused with the egg cell from sheep B. If you look at this now, it is showing us that empty egg cell, empty because the nucleus was removed and thrown away. And now we've got the DNA material, which is the nucleus from where? From sheep A. And this is now a body cell, but not a gamete, not an egg cell or a sperm cell. So they are fused together. And now there will be artificial manipulation. And what will happen? The fused cell develops into an embryo, which is now placed in the uterus of a foster mother. So in other words, now with biotechnology, the genetic material, which is the DNA from sheep A, which is now inserted on the content of the cell without the nucleus from sheep B, it's now manipulated to start growing. And because there must be surrogate parent or surrogate mother, this is now the foster mother. That is where it is going to be inserted. And guess what will happen? This embryo will start growing. And as it grows, then this is now sheep C, which is now our surrogate, carrying the embryo for development. And amazing enough, you now look at lamb is clone of sheep A. Check something here, very interesting, which is gonna make your life easy. If you look at sheep A, what color was it? It was white in color. Look at this little lamb here. It is also white in color. One question that you might be asked by examiners is, what is the, because clones, we said cloning is process of creating genetically identical copy of an organism. On this example that I've told you is that sheep A is exactly the same as lamb A. Right, because this lamb is exactly the same as the parent, but what is the difference? The only difference that you'll notice from cloning outcome is that the cloned individual or organism is going to be young, because now it must still grow, but genetically is the same. That's the only thing. Age is the difference. So you can see all the different steps that are indicated. I used the diagram to take you through the illustration. Once you master that, you will never go wrong. But if you now come to the actual story of how cloning is taking place, then you can say, depending on the scenario that you are going to be given, you don't memorize. It's egg taken from sheep B and nucleus moved and thrown away. The body cell of sheep A is taken and nucleus placed in an empty egg cell of sheep B. An egg is stimulated to start dividing by mitosis. An egg has the DNA from sheep A and will grow into an embryo, and sheep A clone results. There's no way that you'll forget cloning, I reckon. Benefits of cloning. So cloning has got its own good outcomes. Some is that therapeutic cloning can replace damaged tissues, e.g. skin, heart cells, and bone marrow, so helping to save human lives. Genetic diseases could be prevented. Superior animals may be bred to improve food supply and quality. And research in any form improves skills and could open other avenues due to spin-off technologies which could help mankind in the future. You can see that on regular basis there is growth and new developments in technology. So biotechnology is growing field, and there's more of exploring on it. So you may see that cloning is tapping us into the future. I want us to speak about genetic modification in terms of the production of insulin. I spoke a little bit about insulin story. This is now insulin, which is the hormone, which is naturally produced by the islets of Langerhans, which are the special cells that are found in the pancreas. However, people that are diabetic, they need to get help. And the help that they get, they are supposed to get insulin. So if my pancreas or my islets of Langerhans are failing to produce the necessary insulin to regulate, to balance my glucose levels, there must be something from the medical world that I am given insulin and I artificially inject myself, and the insulin is going to now control the blood sugar levels. So unfortunately, in the past, animals were used, pigs were used, and cattle were used to be slaughtered so that they can take all those different insulins for human use. But there were a lot of issues around that. Today, due to biotechnology and due to genetic engineering, insulin can be made. I want us to look at how this is made from the human insulin. So what happens is that there will be two different organisms. So we've got the human cell and we've got the bacterium. So it's bacterium and human too. Just keep a note on that. So what will happen here on the bacterium? We are going to have what we call a plasmid. A plasmid is a round DNA which is self-replicating. You have learned about replication, so this is not Greek to you, that it's making an exact copy of itself. When the DNA makes an exact copy of itself, that is replication. So the plasmid, it's just a round DNA ring that is found on microorganisms like bacteria, and it can be manipulated to replicate itself. And now this plasmid is isolated. You can see now enzymes are used in the process. We can call enzymes chemical scissors, but don't mention chemical scissors. I'm just saying that they will isolate, take the plasmid, and look now. Plasmid is cut. You can see there's a place where the plasmid is cut with an enzyme. And if we look at the other side, human cell, we've got the chromosomes, and you know that insulin is found on the chromosomes. It's genes. And this now sliced up chromosome, you can see the chromosome is now straight. You see the chromosome is straight. And what happens is that there's an isolation. Gene responsible for the formation of insulin is isolated. And what will happen? You can see now the two arrows are taking us to one direction. This is now our plasmid. This is the plasmid. And you can see this is now the insulin gene that is inserted on the plasmid. This now is going to be put in a suitable bacterium. And this is the plasmid that is now inserted on the suitable bacterium. And plasmid with gene inserted into bacterium, you can see now it's there. And due to the asexual reproduction of bacteria, because bacteria, they reproduce asexually through binary fission, you now have, and it's very quick. You can see now that from one, we've got the production of two, and this two will give rise to two more, and two more, which is 4, and 4 more will produce again to give 4 more, 16, and 16, 32, and the number grows very fast. And in that fashion, the plasmid carrying the insulin gene is also replicating and it's reproducing. So there's more plasmid with the attached insulin. Then what will happen is that there's the plasmid which has got the insulin gene from the human chromosome. And for your information, just remember that this is now called the recombinant plasmid because it has now the human insulin gene inserted on it. And we now say plasmid is recombinant, and we say that the bacteria is transgenic. You know, transgenic, we have spoken about it, that it's the bacteria that is now having something, gene that is from a different species. For this instance, it's coming from humans, and then now it's grow in culture, so they culture it and make it more, and look at the end, extract of insulin happens. So here is an injection, and people can just draw insulin from the bottle and they inject themselves should they have insulin related or sugar diabetes or regulation of blood sugar related challenges. Paternity testing, this is now about checking or tracing to see if someone is a parent or not. And here we've got DNA profiles. The process is called DNA profiling. And on this, two things or three or different things are compared. We've got the mother's band that are compared to the child's band to see which bands correlate. And the remaining bands of the child are compared with the possible father. If you look at the example that is given here, we've got the mother, and the mother, yes, mommy, and you can see that the bands are in gray. But here is the child, and there are two fathers, but we're not sure which one is the father. So you can see that when we check the child and mommy, you can see here, the child and mommy, they have got the same bands, they correlate, there's a correlation, there's a correlation. But if you look at the father, let's say this is how you work it out. Check father one. Father one, this bar is standing alone. This band is not bar, but the band is standing alone. And the father, so father one, this is standing alone. It's not matching. And this father is standing alone. But look at father two. So there's the band for the child. This one band is going with the father two. And this band is also going with this father. Then we have got child, let's check, father two, this band is matching with the child, and father two, this band is matching with the child, and we've got father two, this band is matching with the child. So you can see that there's more bands that of father two that's matching with the child, meaning that father two is the actual father of this child that is given as an example. Let us speak about mitochondrial DNA and tracing. This is, you are going to learn more about it in evolution. It's a chapter on its own, but mtDNA, you can see how it is written. It's mitochondrial DNA. It is, you see, you have learned about the cells in grade 10. So you'll see that we've got the mitochondrion, and that's where we find mitochondrial DNA. Very important for us to notice that you see we've got a female and you've got a male. When fusion takes place, when fertilization takes place, this is the whole ovum with all the content of the ovum. However, when the sperm cell fuses, it's only the nucleus that fuses with the nucleus of the ovum. And see this sperm cell, the mitochondrial and everything is discarded. So it's not part of what is there for the developing embryo. And so in other words, they can be the mitochondrial DNA can be traced from males and female, boys and girls, from up to the most ancient female ancestry. I just want you to keep this in mind because this is going to help you when you do the chapter on its own, which is evolution, later on. We've got some activities that we can look at. So this is an extract that we are given. You read it and you read and you read and you keep reading. So the extract is saying the bacterium Bacillus thuringiensis produces a toxin called Bt that kills insects. This bacterium is used to genetically modify maize to contain the Bt toxin. This Bt maize is toxic to insects. The first question. Describe, it says describe. Check your verbs whenever you get a question. How Bt maize is modified to insect resistant. The BT producing gene is cut from the bacterial DNA. You have a mark on that, and then inserted into the maize DNA. And this is now recombinant DNA. I spoke about recombinant plasmid earlier on, and that causes the maize to produce BT toxins. You have given a description of how BT maize is modified. Question 2. Explain 2 reasons why farmers might want to grow BT maize. It's an explaining question. And the reasons that we have is, look at this. We must have a cause and we must have the effect. The cause is that fewer crop damage, which will do what? Which is going to give us increased food. Or we can have food security, we can have healthier food, and many other reasons that we can give. Because fewer crop damage will lead to increased yields. We have more and more food for people and profit, for example. So explaining it's a cause and effect scenario all the time. Make sure you maximize your explanation so that you get all the marks. For this, it was 4 marks and you can get your 4 marks. Another reason is that it reduced need for pesticides. You know, pesticides, they kill pests. When we speak about pests, we have got insects, we can have a lot. We've got insecticides, which are or fungicides. So reduced need for pesticides, and farmers save money or less toxins to consumers or environment. We know that when pesticides are used, they can harm the environment. They will go to the soil, carried by water to the water bodies, algae bloom, and list is endless of all the negativity. And also farmers don't have to spend a lot of money to buy the pesticides to try and fight the pests. We have got a long passage which is now telling us again about GM, genetic modification of crops. Genetic modification of crops began with the discovery that the soil bacterium agrobacterium could be used to transfer useful genes from unrelated species into plants. The gene called BT, which produces a pesticide toxin that is harmless to humans but is capable of killing insect pests, is one of the genes most commonly inserted into crop plants. Many new GM crops such as maize, potatoes, and tomatoes are modified to be pest, disease, or weed killer resistant. GM foods could have unforeseen effects. Toxic proteins may be produced, or antibiotic resistant genes may be transferred to human gut bacteria. Modified crop could become weed killer resistant or super weeds. Modified crop could also accidentally breed with wild plants or other crops. Question 1. According to the passage, how did genetic modification of crop begin? You go back to the passage. Do not read the passage once. You read and you read again so that you remember and you go and refer. It began with the discovery that the soil bacterium agrobacterium could be used to transfer useful genes from unrelated species into plants. It's there on the passage. Question 2. Explain why a plant which is modified to be weed killer resistant could be a problem for farmers. Why? Because modified crops may become super weeds. Super weeds, there's nothing that you can do to get rid of them. It's a stress to really try to fight super weeds and breed with other plants to become super weeds. They are difficult or they are expensive to kill and could outcompete the original crop. So the indigenous crops can be outcompeted by those ones that we thought were trying to come up with a good solution. Question 3. Give 2 examples in the passage of the use of GM crops that may be a potential threat to human health. Again, you go back to the passage and you are going to give the answers from the passage. And the answers are: toxic proteins might be produced and antibiotic resistant genes may be transferred to human gut bacteria. So you think I'm getting this from somewhere? No. It's coming exactly from the extract. You can see, yeah, toxic proteins may be produced. And the other part of the answer is antibiotic resistant genes may be transferred to human gut. Easy, straightforward. But right there, and you get your marks. Now we're talking about cloning. The first cloned animal in Africa, a calf named Futhi, was born in Northwest in South Africa on 19 April 2003. No fertilization was involved in the production of Futhi. She was produced from a single cell taken from the ear of donor cow named LMJC 865. The donor cow had a high average milk yield of 78 liters a day. Cloning allows for the production of organisms with desired characteristics. Some people argue that cloning reduces genetic variation in the offspring with no further genetic improvement. Cloning is an expensive procedure and may not be economically for commercial agriculture. The first question, according to the extract, state one advantage. You go to the extract, you give us one advantage of cloning. And what is the advantage? It allows for the production of organisms with desired characteristics. It's there on the extract. And you give us one disadvantage of cloning. And what is the disadvantage? It reduces genetic variation in offspring and it is expensive. And the next question 2, state why the donor cell was taken from LMJC 865 and not from any other cow. Why specifically this cow? It is because it had a high average milk production. The purpose was to get more milk every time. State why an ear cell was used and not the ovum. Why? Because we know a diploid cell is an ear cell. It has got genetic information that is needed, and an ovum is haploid and only contains half of the genetic information. And question 4, briefly describe the process of cloning. And we all know the process, which I am not going to go through. We have learned about the process of cloning. I've explained fully. This activity is about paternity. So when we do paternity testing, we've got a thief here. I am not going to read for you, but I'll give you a background. A thief had a broken glass, and what happens? There are 3 suspects. For you, what does the diagram represent? It represents a DNA profile, but not DNA profiling. Which suspect is likely to be the thief? Definitely the thief is Q, and give a reason for your answer. All the DNA bands match with the DNA of the blood. So you check Q, you'll see the bloods that are found. There's a match there, there's a match there, there's a match here, there's a match here, there's a match here, there's a match there. And this activity is talking about one adopted child. So which of the children is adopted? You see, Tom, it's the father, Maria and Mary. Which one is adopted? And the adopted one is definitely Mary. Explain your answer. And the explanation is there are no matching bands. You can see if you look at Mary, she does not match with Tom and Maria. And we've got the summary. We have done all this, so you can look at the summary of what we are supposed to know about mutation and genetic engineering. On the 8th of May, please be tuned up for NEVA's coordination. Let's call it a day. Thank you.